Good morning and good afternoon to everyone. Welcome to presentation session A2 diagnostic tests. My name is Kate Dupnick, and I am joined by Professor Richardis to co-chair the session on diagnostic tests. We have 6 abstract presentations, progress reports, which I'm very excited to hear about. There will be 8 minutes for each presentation, followed by 6 minutes for questions. If you have questions, please put them in the Q&A box. And we're looking forward to hearing about progress and having some discussion. So our first speaker is Ms. Faria Hossain from ICDDR B. And I will let you share your slides, Ms. Hossain.
Hello and good evening from Bangladesh. I am Faria Hossain and I'm here. I'm working at the International Center for Diabetes Research, and I'm here to present my progress of my present the progress of my project, which is entitled the Recombinase Aided Amplification Assay Incorporated in Mobile Suitcase. So here is a brief overview of the current status of leprosy in Bangladesh. Bangladesh is one of the 22 global priority countries for leprosy elimination. Although the country has achieved the WHO elimination landmark back in 1999, as you can see from the trend that the new case detection rate became static after that. And for last 10 and for the last 2 decades, the range is between 3,000 to 4,000 new cases every year. And 6 to 8% of them are suffering from grade 2 disabilities. And 5% of them are child cases. Okay. So currently, leprosy in Bangladesh is diagnosed solely based on clinical evaluation, based on the characteristics of skin patches and with sensory loss. The slit skin smear, a microscopy with a slit skin smear is recommended, but due to, and it is obviously the gold standard, but due to its low sensitivity, it is not really very much utilized. So we are considering other tests like serological tests, but the serological tests like ELISA and lateral flow assays have these limitations of low positivity for PV leprosy, although they are very good in MB leprosy. And finally, there are molecular tests like PCR and real-time PCR. They're both comparatively highly sensitive and specific compared to the serological and tests and the microscopic tests for both PV and MB leprosy. But it is costly and requires referral laboratory infrastructure. Therefore, it is not always accessible at the resource limited settings. To address this gap, we have developed isothermal nucleic acid test, which is a recombinase aided amplification assay targeting the RLP region of the Mycobacterium leprae. It is rapid, feasible, and cost-effective compared to the PCR. And the limit of detection is 11 DNA copies as per our analytical results. And we observed no cross-reactivity across 20 different organisms, including the Mycobacterium species. And we have combined this assay with a rapid extraction method, which is magnetic bead extraction method. And finally, we have incorporated the assay into our solar-powered mobile suitcase laboratory. As you can see, there are 2 suitcase laboratories. One is designated for the extraction, and one is for the detection. The extraction suitcase lab contains every equipment necessary for the rapid extraction of the DNA from the organism, and the detection suitcase incorporates the T8ISO instrument for the detection of the DNA, and the suitcase labs are powered by the solar panels, And also they function with direct electricity as well. And because they are mobile, because they are easily transported, can be easily transported to the remote areas. So the suitcase labs, and when incorporated with the recombinant agent amplification assay and our rapid extraction method, it can be used to deliver the service at the point of need, and can be applied at the field setting as well. So here is a brief overview of the whole procedure. As you can see, the rapid extraction method is very simple and includes only 3 steps. In the first step, we mix the sample with the magnetic beads and the lysis buffer. And the next step, we heat it for 95 degrees Celsius for 10 minutes, and then they are set in a magnet for the reverse purification, and the resultant supernatant is ready for use to detection. And for the assay, we use these lyophilus reagents, which can be stored at 40 degrees Celsius for 3 months and is very much stable. The whole test to result takes only 30 minutes, 15 minutes for extraction and 15 minutes for detection. So the objective of my study is to determine the diagnostic sensitivity and specificity of the assay for detection of leprosy in clinical samples, and when compared to the real-time PCR and slit smear microscopy. For the real-time PCR, we are actually using 2 systems. One is the CSX96 real-time PCR system, and another system is the mini-qPCR. This mini-qPCR device, which is very compact, and you can see it can be easily set into the mobile suitcase laboratory as well. It requires 30% less time for detection compared to the CSX real-time PCR. So our study is a case control study. We are aim to enroll 50 MB leprosy cases, 50 PB leprosy cases, and collect the skin smear, skin punch biopsy, and nasal swabs from the cases to test finally with the microscopy, MLPCR, MLRA, and mini-qPCR. Additionally, we are also collecting the 200 household samples from 200 household contacts, disease controls, endemic controls, and non-endemic controls. We will be collecting nasal swabs from all the controls, except for PKDL. We will use the PKDL skin punch biopsy, carbon extracted DNA from the PKDL skin punch biopsies, and will be evaluated through MLPCR, RA, and mini-qPCR. So we define the cases according to our national guidelines and technical manual on leprosy of Bangladesh, and we have classified the cases in multibacillary and postbacillary leprosis according to the definitions of WHO. We also here you can see the definition of household contacts are the household members of each confirmed patient with no signs or symptoms of illness. And for the disease control, we have used GenExpert and culture positive tuberculosis patients and PCR confirmed PKDL patients. For the endemic control samples, we have recruited healthy participants from the endemic regions of leprosy, not living within 20-meter radius of the household of the patients of the cases. And we also enrolled non-endemic healthy controls from the low prevalence area for leprosy. So far, up to March 2025, we have enrolled a total of 41 MB leprosy, 39 PB leprosy, 115 household contacts, 30 endemic controls, all the disease controls, and 25 non-endemic controls. So here are our results, first results. We have performed the microscopy in 35 PB leprosy cases, and all of them were negative, and 36 of the MB leprosy cases, and only 13 of them were negative out of 36. And we have tested 20 PB and MB leprosy cases with the MLRA assay in all 3 types of samples, and only one nasal swab sample was positive in PB leprosy case, and whereas 8 6 and 5 cases were positive out of 20 in skin punch biopsy, Christmas smear, and nasal swab for MB leprosy cases. And all other household contacts and controls were negative. However, we have completed the PCR assays, the MLQ-PCR and both the MLQ-PCR and the mini-qPCR assays for 36 MB leprosy cases and 35 PB leprosy cases, and analyzed and the primary analysis was done. As you can see, we both the assays, the skin punch biopsy was the highest, showed the highest sensitivity for both MB and PB leprosy. However, the overall sensitivity of was higher in MLQ-PCR, which is 72.45%, and compared to the mini-qPCR, which is 54.54%. Okay. We have also calculated the specificities, and none of the samples and none of the controls were positive in both of the assays. So we found a specificity of 100% for both the assays in all the controls. The challenges we are currently facing is the lower sensitivity of the MLRAs in clinical samples. I think we need to perform we need to explore and perform the whole genome sequencing of the local strains to identify the possible sequence variations within the target site, because the target we selected was from the GenBank and it was universal and Leprosy strain. So we really think it is necessary to sequence the local strains. If there, so we can decide on the revision of the primer probe. And finally, furthermore, we have detected 8 MB and 1 PB case, which were negative in MLQ-PCR and mini-qPCR with all kinds of sample types, and as well as in SSS microscopy, which actually raises the question if they were actually correctly diagnosed clinically. So there is a need to follow up such patients for treatment responsiveness after 6 months and 1 year for PB and MB. And also, we need to test the samples for other endemic skin entities like PKDL. So our next steps includes the patient to complete the patient enrollment, sample collection, and testing the rest of the samples. And we aim to complete our data analysis and prepare our first draft manuscript as well. I want to thank my colleagues from ICDDR B, our collaborator, Dr. Ahmed Abdul Wahid from University of Minas Gerais, and the INF Foundation and the Leprosy Mission Bangladesh for their continuous support. I also want to thank the Leprosy Research Initiative and Turing Foundation for the funding support to perform this important project and important and timely project. Thank you all.
Thank you.
We have a number of questions, and the first is very practical. What is the cost of the suitcase for extraction and detection?
The cost of the suitcase is the total system is about 7,000 USD, including all the equipment.
Yeah.
But excluding the data instrument, which will cost additional 5,000 USD.
Yeah. Can you explain, that's the second question, the detection principle of RAA? Did you perform a limit of detection? And if so, how low numbers of M. leprae can you detect?
As I have shown in my slides that we performed the analytical sensitivity, and it was 11 DNA copies from M. leprae genome we could detect in our analytical sensitivity assay.
Okay. And another question is about the location of the skin biopsy. So from which location was the skin biopsy taken?
Okay, the skin masses were taken from different locations actually. We selected the locations based on the active lesions, which were expanding, and therefore the probability of the detecting the Mycobacterium was higher.
Yeah. And a question also about the skin biopsy is how is it prepared to be crushed in the tube for DNA extraction? So what is the technique for skin biopsy preparation?
We collected the skin biopsy directly in our buffer, and then transported it, and then extracted it. So it was pretty simple.
Okay, thank you. How long does it take, I guess, with regard to the suitcase setup, how long does it take to generate results?
Okay, it takes 30 minutes, 15 minutes for extraction and 15 minutes for detection.
Okay. And are you actually already applying the suitcase, or is it still the comparison within the lab, with the suitcase in the lab and your lab setup?
Okay, actually, the suitcase lab is already applied for detection of leishmaniasis, and dengue, which were evaluated, and the phase 2 diagnostic trial was already completed. And in this study, we have developed this leprosy assay, and in this study, we actually, well, it is the first case control study, so we are still evaluating it. We are evaluating its diagnostic efficacy, and we'll find out any troubleshoot any problems here, and maybe we can improve the assay to detect it finally. So it is not applied for leprosy yet, but it is well, it has been applied for other diseases.
Thank you. Probably we're reaching the end of our session. There are still some questions about the clinical leprosy cases and not being able to detect with PCR. So maybe we can come back to that at the end of the session, because I think those are important questions why you are not finding all the cases through your techniques.
Especially with a lower limit of
Okay.
detection of 11, it would be surprising to not detect it. So the genetic the genetic profile of your local strains is an interesting approach. But I agree with Dr. Richardis that we can table these questions till the end when we have time for group discussion, and maybe this can play into some of the other discussions about diagnostics. So thank you so much for presenting your work, Ms. Hossain. Our next speaker
Thank you.
is Ms. Louise Pierneef from Leiden University Medical Center. Ms. Pierneef, if you want to share your presentation.
Okay, hello everyone. My name is Louise. I am a PhD candidate in a group of Professor Geluk at the LUMC. For the past couple of years, I've been involved in this project on point-of-care tests for leprosy in South America. So for many years, our team has been working on identifying host immune biomarkers for MB and PB leprosy. And we try to include those in field-friendly diagnostics. And what we found is that for MB leprosy cases, we can actually detect most of them using antibodies against PGL1, which is here shown in blue and green. But for PB cases, this is not the case. So there you would need additional markers, such as cytokines or chemokines. So the combination of markers can actually improve the diagnostic potential for MB, BT, and PB leprosy. So in this study, our first aim was to evaluate a quantitative point-of-care test for the detection of M. leprae infection. The test you can see here, it detects anti-PGL1 IgM, and it is now also available in a biodegradable format, which is also recently published in Infectious Diseases of Poverty. So if you want to read more about this, I refer to this paper. But in this study, to evaluate this test, we collected 20 microliters of fingerstick blood. This was mixed with buffer. It was loaded onto the test cassette, and then it was scanned in a portable reader, which can also be battery operated. So this was all done in the field. Then as a second aim, we wanted to test host biomarkers for leprosy that were previously identified in an Asian population in Bangladesh, but we wanted to see whether we could also apply those to the South American setting. And for this, we took whole blood, we performed a whole blood assay, we tested multiple cytokines and chemokines, and we scanned everything and did the analysis at the LUMC. Before I go to the results, I just want to highlight some challenges that we encountered during the project. So the project started in 2021, which was still during COVID, and this made it sometimes a bit difficult to actually go through with the recruitment. So and also in Bolivia, we found that cases were actually scattered over a very large geographical area. So sometimes to actually reach one case, we had to drive for 6 hours. And therefore, with all this traveling, it was a bit difficult to reach this within the project budget. Also, there was a lot of migration going on, both from cases, but also from the local health workers that were involved in the project. And also shipment of lab materials from the LUMC to sites and the other way around became more difficult due to stricter regulations. In Brazil, it seemed difficult to confirm suspect new cases, and there were also some difficulties in continuation of the sampling after the project teams left the site. And also there, the shipment of the materials from LUMC to sites and the other way around became more difficult. However, of course, we try to mitigate this, and therefore in Bolivia, skin and health camps were organized to increase the numbers of participants. And in Brazil, there was a second mission organized to the high endemic area in the Prata. And we also included an additional dermatological hospital for recruitment of cases in the non-endemic Curitiba. So first, some results from the anti-PGL1 IgM fingerstick blood test. Although the new case numbers are not that big, still we found that there were increased levels of anti-PGL1 IgM in new cases compared to endemic controls. And if we look at those individuals displaying the highest anti-PGL1 IgM, we also found that they had very low RLAB-Q PCR CT values. If we look at seropositivity, we see the highest in MB cases, then PB, which is as expected, and then contacts also had higher seropositivity as well as anti-PGL1 levels compared to endemic controls in this area. So this confirms what we have found before, that anti-PGL1 is associated with the bacterial load, and we found higher anti-PGL1 IgM in contacts than in endemic controls. Then the results from Brazil for this test. From the high endemic area of the Prata, we had 10 new cases included, but again, we found increased anti-PGL1 IgM in comparison to contacts. If we look at the seropositivity there, we see 75% of MB cases and 0 of PB cases. And here we have combined contacts and endemic controls, because this area is actually a sort of hyper endemic area. So everybody is actually in contact with someone who has leprosy. Then we also included the additional non-endemic area in Brazil. And also there we find increased anti-PGL1 IgM levels in new cases compared to contacts. But what we also see is that in a high endemic area, there is higher seropositivity in the contacts and endemic controls compared to the contacts in the non-endemic area. Then if we look at the other markers, first these are the results from Bolivia, we find increased levels of IP10, SA1, A2, and CRP in new cases versus context, although the new case numbers are quite low. And then if we combine those significant markers with anti-PGL1 IgM in sort of a 4-marker score, so calculating the number of positive markers per individual, we actually see a very good discrimination between the two groups, so the new cases versus the contacts, with an area under the curve of 0.99, sensitivity of 100%, and specificity of 90.5%. Then for Brazil, we also tested those same markers, and we found significantly increased levels in new cases of IP10, SA1, A2, S100, A12 next to anti-PGL1 IgM. And then again, if we combine the most discriminatory markers into a 3-marker score, we find a very good discrimination between the two groups, with an area under the curve of 0.94, sensitivity 84%, and specificity of 97%. So with this, I hope to have shown you that there is added diagnostic value in combining multiple biomarkers into a signature, and such combining those 3 or 4 markers could actually be incorporated into a cassette format, which you see here, which is very applicable to the field. And why is this important? So we found now that biomarkers that were identified in an Asian population could also be applied to the population in South America, although the analysis is still ongoing. We did endemicity and case mapping. Capacity sharing was done. A PhD student from Brazil joined our lab for 6 months, and the qPCR for our lab was set up in Bolivia. We have created more awareness, and we have more information on infection rates now. And we also are evaluating the test feasibility and acceptability using a questionnaire among health personnel, but this is still ongoing. So I would like to thank the LUMC team in the test development, and also, of course, the field teams from Brazil and Bolivia. And thanks to all of you for listening. And if you have any questions, then please let me know.
Thank you. Thank you very much.
Okay, thank you. Very interesting study. We already have one question coming up. For markers like CRP, did you rule out any other infection?
Sorry, for CRP, you mean?
For markers like CRP, did you rule out any other infection?
No, but we are not, of course, we are not looking at CRP alone. It's always in the context of also, of course, clinical signs, but that's also why we use the combination of biomarkers and also with a specific one like PGL1.
Okay, thank you. I don't have any other questions. How are we with regard to the time? Otherwise, we move on and we can come back to you at the end if more questions arise.
We have a little bit of time. Could I ask a question about the acceptability, sort of with the finger sticks? That can be a sort of a painful experience. What was the thought of that? And is this something that could be readily adapted to a regular phlebotomy?
Well, actually, we didn't really experience a lot of refusal. I think, yeah, finger prick blood, it's actually quite low invasive, especially compared, of course, to biopsies and slit skin smears. So we did not really have a lot of refusal regarding this.
Is it something that could be applied to phlebotomy, like just a regular venous blood draw?
Yes, but this is, of course, the finger stick blood. It is, we chose this because it is both low invasive and also because, of course, because of the field friendliness and the that it needs to be a rapid test. So I'm not sure if that would, yeah, increase the field friendliness and of the test.
I think there might
There's
be.
one more question.
I think we have time for that one, and then we can move on.
Okay.
So the question, second question is, did you analyze any individual responses to all the biomarkers?
You mean if we looked at one individual comparing all the biomarkers? Or...
Yeah, I think that's the question, and if not, maybe Sunita can elaborate. If it is possible for one, would it be positive? If it's positive for one, would it be positive for all?
That is not always the case. Sometimes we see correlation, for example, for CRP and SA1A2. They usually correlate, but of course there's also strength in not always having the same markers being all markers being positive, because you want added diagnostic value of the combination of your markers. So it would be good if one would test positive, negative, and then another one tests positive. Then you actually pick up more cases. So.
Okay, thank you. There's one more question. In describing your challenges, you mentioned that it was difficult to confirm new cases in Brazil. Can you explain why?
I think this is because the expertise maybe is diminishing. So yeah, we actually did really experience a lot of trouble with that. Sometimes it was only after maybe a year, and then they were followed up again. But then if you have sample from a year earlier, then you're not sure whether they are, they might have been maybe a PB case, self-healed. So in the end, what we did is we went back to suspect new cases. Then when their diagnosis was confirmed, then we took a new finger stick blood sample or whole blood assay sample, just to make sure that we had the sample at time of diagnosis.
Okay, so it was a matter of difficulty of the clinical diagnosis.
Yes, yeah.
Okay, thank you.
Thank you very much. Okay, our next speaker is Ms. Lina Restrepo-Rivera from Instituto Colombiano de Medicina Tropical. And Ms. Restrepo-Rivera, if you could share your presentation.
Yes. Good morning, everyone, and thank you all for being here. Today I'm going to present a project evaluation of 5 transcriptomic biomarkers for leprosy in a Colombian population. To start, in 2023, over 180,000 new leprosy cases were reported worldwide, and 30% of them in Latin America. In Colombia, my country, nearly the half of the municipalities have reported at least one leprosy case in the past 7 years. And some high-burden municipalities reported even 20 and 40 cases. This is a public health concern, and new biomarkers is needed to support the diagnosis. One of these biomarkers is HES1 from Notch signaling pathway. Several studies have shown that HES1 is reduced in skin biopsies from leprosy patients in several structures like eccrine glands, hair follicles, and epidermis. This opens the possibility to use less invasive samples like skin scraping to analyze if HES is reducing these structures. Another potential biomarker is RIX4-LEP. RIX4-LEP is a signature that composed that for genes. It was a result of one study of Dr. Geluk, and analyzing gene expression in whole blood, they can predict which household contacts are likely to develop leprosy. So with this information, we ask, are there changes in risk for LEP and HES1 signature in Colombian leprosy patients? And to answer this, we have performed field trips at the moment in 20 municipalities across the country. And then we have performed clinical evaluation in leprosy patients and household contacts. To answer the question for risk for LEP, we aiming to gather 130 samples, whole blood, in packaging tubes that preserves the blood's RNA. And in household contacts, we aim to gather 1,000 samples, including PAX genes, PGL1 cassettes, and WBA vials. To date, we have collected and evaluated 104 patients, 78 of them were male, and around 80% with multibacillary leprosy and 60% with lepramatous leprosy. And 370 household contacts have been evaluated. 230 were female, and mostly of them report daily contact with the index case. From these contacts, we have found 82 individuals with symptomatic in skin or peripheral nerves, and 8 of them were positive to leprosy by qPCR, 5 individuals, and 3 through histopathology, and they were reclassified as patients. Last year, we sent to Leiden University a batch of these samples, 63 PAX gene tubes from patients, and 255 samples from contacts, including PAX gene tubes, PGL1 cassettes, and WBA vials. We have partial results from this. Notably, 5 household contacts show high levels of PGL1 antibodies. And this is not a test to predict leprosy, but it's very important. It's a useful tool to identify which individuals need a closer follow-up. And to answer the last part of the question for HES, we perform epidermal scrapings using dermatological curettes. We aim to achieve, our goal is to achieve 65 samples from MB and other 65 for PB, and to compare this through RT-qPCR with healthy controls and differential diagnosis. After sample collection, RNA is extracted using a commercial kit, and we have standardized RT-qPCR using a one-step. And these are some of the results. Here you can see the target gene and the curves from serial dilutions and standard cubes in this PCR. And of course, we have also standardized the housekeeping genes as well. And in this case, for HES1, we yield 93% of efficiency. At the moment, we have collected 32 epidermal scrapings from patients, and 10 of them are new patients, and most of them are MB. As well, we have gathered 12 controls and 9 differential diagnoses. This is an example of a PCR from an epidermal scraping sample from patient. In pink, you can see the housekeeping gene, and in green, the curve for HES1. What's next? So we need to follow the RT-qPCR analysis using the other epidermal samples and the follow-up of household contacts and symptomatic individuals. As well, we need to do the analysis for risk for LEP at Leiden University and continue with the field research. We face some challenges for HES1. Collect the number of samples and to collect new patients could be difficult due to the reduced report of new cases in the country, and as well ongoing public order situation that we face. And for risk for LEP, it could be difficult to collect the 10 the 1,000 samples from contacts, it be challenges. And of course, the limit time for the follow-up that we can maybe to identify leprosy progressors. And of course, we need to determine the diagnosis of the other patients. And we want to thank to Leprosy Research Initiative and Turing Foundation and our team. Special thanks to Dr. Galuk and Luis from Leiden University, Dr. Mieles from Sanatorium, and our local team, Dr. Nora, Angeli, Danilo, and of course, Hector. And thanks, of course, patients, families, and our allies.
Working with communities have been transformative. Listening to leprosy patients and their families has helped us to understand their dreams and challenges. For example, they face difficult times when they received the leprosy diagnosis and started treatment. However, they have shown incredible strength and resilience. They find the greatest motivation to move forward in their family. For this reason, they become our inspiration to keep working on leprosy.
El hecho de tener a una familia donde en algún rincón hay personas que ellos saben que es lo que quieren y si hay una cura para ellos. El hecho de tener varias se da a una orden de cabeza, pero el objetivo más que explícito es que estoy indicando.
And thank you.
Yes, that's very inspirational conclusion to your presentation. So thank you very much. While we wait for questions, and I encourage all the participants, if you have a question for Lina, please put it in Q&A. But I can start with a question of my own. It's just for clarification. At the beginning, you distinguish between patients that are MB and patients with multibacillary leprosy. In my mind, they are more or less the same, but can you explain from your perspective what the difference is between these two categories?
Can you repeat the question, please?
So my question is, you distinguish between MB patients
Yes.
and patients with multibacillary leprosy.
Okay.
In one of your first slides, what is the difference? Because in my mind, they are very similar categories.
Okay, maybe there's a mistake for my part, but it's the same.
Okay.
Yes.
There was a slide, maybe this is what you're referring to, where
Yeah,
you
one of your
said
first slides, maybe
multibacillary
it meant
and
to
lepromatous.
say MB and PB.
No, I think that it was, I wrote it down to 85% multibacillary and then 60% with lepromatous forms. So I was assuming it was like the LL pole of multibacillary, but
Okay. The
I'm not sure if
matters
that's,
of, yeah, but it's a matter of classification according to the, yeah.
I think sort of it's interesting that of your 104 patients, 85% of them were MB. And if 60% are actually polar lepromatous, that really speaks to a very like a underdiagnosis issue.
Yes.
And these sort of the voices of the people who are affected by leprosy that we heard at the end really speaks to that sense of long-term symptoms and waiting for an answer. And so it's kind of probably just the beginning for the outreach and intervention.
Yeah, that's the reason why we are doing this firstly, because in our country we face many difficulties to reach remote areas, and in those remote areas are patients that face misinformation and even stigma that complicates a lot the diagnosis and early diagnosis. And of course, we have the difficulties with professionals that have not enough expertise and maybe, to classify clinically those patients at time. And maybe they pass several years of months waiting for a certain diagnosis, and this is a difficulty.
So how do you envision these diagnostics being integrated in your health system with those challenges of delivering care in remote areas? Do you see it as being something where there's a central lab and samples are sent in, or point of care? And then I think after this, there's a question in the Q&A.
Okay. In Colombia, in some remote areas, some samples takes a lot of time to reach a laboratory and have a diagnosis, and mainly is clinical the diagnosis, and bacteriological or histopathological diagnosis could be difficult due to some areas are very remote, and maybe in some areas even have the opportunity to take a sample to send to the laboratory. So yes, in the health system it could be a priority, but maybe we need to continue working on that in Colombia.
There are two questions now. The first one is from Dr. Bishwa. Given a non-specific expression of S1 across tissues, what makes it a promising marker for leprosy-specific pathology?
Okay. Okay. S1 in biopsies have been shown a reduced expression or marker, but we have working on that, comparing with other diagnoses, and is a marker for inflammation, and we know that S1 is reduced in other pathologies that compromise inflammation in those tissues.
Okay, thank you. And do we have time for the second question? Is from Dr. Becky. From your research and preliminary data, do you expect to find one or the other targeted markers or a combination to improve diagnosis?
Okay, currently we are focused on S1 from epidermal samples, and maybe with the whole blood samples that we send to Leiden University, we can explore with those RNAs and other markers maybe for Colombian population that could be specific, but it is only an idea. We need to focus on those 5 biomarkers at the moment.
Okay, thank you. Those were the questions we had.
Great. Thank you so much. Yeah, thank you so much for your presentation and for thoughtful consideration of all of our questions. And thank you everyone for contributing. Our next presentation is by Dr. Frederico Soriani from the Federal University of Minas Gerais. And Dr. Soriani, if you could share your presentation with us.
So, good morning, everybody. It's a pleasure for me to be here to show you some of the results we already have and the project. The project is seeking for immunological susceptibility markers for leprosy. As you all know, leprosy still have a huge number of new cases every year, and Brazil is the second one in the world with in terms of number of new cases. So after the exposure of leprosy, you also know we have natural resistance or symptomatic clearance in some patients, but we also have the development of the disease, symptomatic leprosy, that will vary between two different poles according to Hidley-Joplin. So we have a Th1, the tuberculoid pole, and a Th2 response for the lepromatous pole. And these immune response at the end will contribute very much for the outcome of the infection. So taking this into account, we performed a transcriptome and a miRNA sequencing for all the, I'm sorry guys, for the 5 clinical forms of the disease and controls, and we were able to identify a lot of messenger RNA genes, more than 4,000 genes, and microRNA genes. Actually, our analysis demonstrated that we have a discriminatory profile of the expression of those genes and the pairs microRNA and mRNA that can indicate that the control of the immune response can be done by those microRNAs. So, taking into account the differential expressed genes and the differential expressed microRNAs in the clinical polar forms of the disease, I mean exclusive pairs for the clinical forms, our main question was the identification of the host genetic factors for leprosy susceptibility in a multicentric approach study in the Brazilian population. So to get these results, we analyzed all those genes that I mentioned before, but we analyzed them in pairs. I mean, the epigenetic controller, the microRNA, and the target, the mRNA. This allowed us to identify opposite expression profiles. I mean, up-regulated mRNA and down-regulated microRNA, or the opposite. And then, analyzing these pairs, we were able to identify in tuberculoid poles and lepromatous poles different pairs. I mean, 42 pairs for lepromatous and 2 pairs for tuberculoid poles. So these genes, these pairs, I mean, microRNA and target, were allowed us to identify canonical pathways, I mean, in terms of enrichment of those genes, even for the lepromatous leprosy or the tuberculoid leprosy poles. These pathways, these canonical pathways that were enriched, gave us a list of different genes, and we took those genes to understand which microRNA were acting on those on the untranslated region 3 comma, and maybe if our question or our hypothesis was true, variations in the seed sequences of those microRNAs in the target genes could represent a different expression profile in those genes and the outcome of the disease. So here I show you just 3 of them. The list is very big, and I'm going to focus on this addition G protein coupled receptor, G2. This is the microRNA that targets this gene in the 3, UTR region, and there is a SNP in the seed sequence of this microRNA. We did this for all the genes, and with the purpose to understand the function or the variation of the expression of the gene based on the ligation or the binding of the microRNA or not, depending on the SNP in the seed sequence. So I don't want you to understand all this tables, just a representative. We did, the first step was to identify the frequencies of those alleles. I mean, the reference allele and the alternative one. Here I show you just for one SNP. We took all the databases around the world already published. And here you can show that the alternative SNP has a extremely low frequency. This would challenge us to get those results from the population and from the genotyping of the population, and it made us to move for a new approach in a large scale analysis. We are doing this right now. Trying to get more genetic markers or more genetic information regarding to those sites and another targets and so on. To do this, we got from a lot of partners in Rio, Sergipe, and Minas Gerais, 3 different endemic areas in Brazil. And we got paucibacillary and multibacillary patients together with household contacts. Here is the number of samples we still have. And the next steps of our work is the genotyping we are already doing in this multicentric approach for trying to understand and make a genetic association studies for the identification of those genetic risk markers for leprosy per se and for leprosy polar forms of the disease. Of course, this was done in collaboration with lots of federal universities in Brazil and research institutes. And I would like to thank all the financial agents and thank you all for the attention.
Thank you, Dr. Soriani. Thank you for your presentation, and there's certainly always a surplus of data when you do these large-scale assays. And can you expand a little bit on the targets where you started looking at the SNPs? Were those with the microRNAs or the messenger RNAs? And does the rarity of the SNP variants in the population that are, in public databases, what is that, for those of us who are not, don't know what that means, what can you tell us why that's significant?
Okay, yes, thank you for the question. The problem is that this very low frequencies of those SNPs in different populations around the world may represent that we will have to analyze thousands of patients in order to get some result back. And this is quite challenging for us. So we moved the approach for a large scale one that can bring us maybe this information for these SNPs, but we are trying to understand, to check if we can get the results for the mRNAs that are targets of our microRNAs. Even if I can't get the SNP in the seed sequence of the microRNA binding site, I can get the target. I can get the SNPs in the targets. So I can correlate them and then make specific experiments for the microRNA in different populations. So we are trying to open our research and then come back.
I think it's interesting because we're now in sort of like this polyome world where you're not just talking about gene expression of protein precursors, you're talking about microRNAs, which are regulatory, and you're also talking about human genome studies to look at the SNPs. And so you're integrating, I think we had some questions come in, so I'll let Dr. Richardis go address those. But I think, starting to look at genome-wide scans in addition to the lesional level gene expression. So that's it's a lot to parse.
Yes.
Yes, we indeed have 3 questions. I'm not sure if we have time to answer all of them, but the first one is about whether you use GWAs for analysis of this gene, or do you target the variants that you show in the table?
Yes, actually, the GWAS will bring the information of different variants in our analysis, and then we can move back, as I told you before, we can move back to specific genes that we already have in our analysis, and even in the main sequence of the gene or in the seed sequences of the binding sites. I don't know if I got the answer, but
well, thank you. Maybe the person who is asking can contact you separately to get maybe some more detail.
okay.
There's another question about miRNA targeting. Could this be the next immunotherapeutics in leprosy?
Very good question. Actually, I'm not sure about that because microRNAs are extremely promiscuous in terms of which gene they are targeting. So we have different binding sites and in different targets. So at the end, the expression of some gene will be a combination of a set of microRNAs that can modulate the expression of it. Even though we still have the relates literature that some microRNAs could influence positively the expression of the genes by another mechanism. So it would be, I don't know about immunotherapeutics, could be with microRNA, could be an approach. I'm not sure about that.
Okay, thank you. One more question is about how specific are the responses of SNPs to leprosy and not general. So what is this? How specific are they?
Yeah, it's a kind of that I said before in the question before. Yes, they are general. I mean, the microRNAs are general for different genes, but if we are talking about genetic approach, I mean the SNPs, trying to understand the function. So the expression of those genes that will function in controlling the immune response or controlling some biological response. So I think it could be specific. At least at the end, the expression results we still have for microRNAs, they are related and they are specific for each one of the 5 Hidley-Joplin clinical forms. So it made us hypothesize that it could be a detrimental control for the gene expression and the outcome of the disease or the immune response.
What are your controls?
Okay, they are non-consanguineous contacts. Okay.
Okay, thank you. Are we good, Kate? Or...
Yeah, I think we should probably continue on to the next presenter. Thank you, Dr. Soriani, for your presentation and consideration of questions. There are, I think, a few more questions in the chat. Maybe we can circle back at the end. And our next speaker is Dr. Suraj Maharjan. Dr. Maharjan, can you share your slides, please? Dr. Maharjan is from the INF Green Pastures Hospital.
Good evening from Nepal.
Thank you.
It's almost late evening here. I'm glad to present my research project, which is under Research Capacity Strengthening Grant, and which is ending this year, September of this year. I'm very privileged to present on the progress. So I'll start with a quote from Paul Brand. There is no better treatment than if the problem had not been allowed in the first place. So we can see I've kept a picture of a thermography picture of bilateral hands of one of the affected leprosy patient. Just to start with the background, early diagnosis and nerve function impairment. And early diagnosis of leprosy, it's we all know it's a challenge, and there's been studies that patients with subclinical neuropathy were not evident during testing of BMT-ST. And also nerve function impairment present in diagnosis is a strong predictor of those immunological reactions or nerve function impairment leading to disability. We've seen significant studies on electrophysiology studies, nerve conduction studies, and autonomic nerve function test in those who didn't present with clinical symptoms of leprosy. So in other study, early diagnosis and prevention of nerve damage is a challenge in leprosy, despite of having multiple assessment tools being available and use of cardinal signs. So let me get the pointer. So we all know that we rely on basic diagnosis mostly with cardinal signs. If not, if we have any lab setup, we can go for a skin or nerve biopsy, or we've talked a lot about serology, PCR tests and all, which is available in very few centers. So we also rely on somatic function tests, sensory and motor, which is more on dependent on patient's feedback. Motor is more dependent on patient's effort and examiner's assessment, how they interpret. But the bigger picture about autonomic part is it's independent of patient and examiner's subjectivity, which is shown in a couple of papers here. So there's use of infrared thermography in many different fields, or also in medicine, as in cancer study, or in skin disease, in plastic surgery, as a surgeon, I say flaps. Now also in peripheral autonomic dysfunction. Also, it's widely used now in diabetic neuropathy. We basically know that impairment of skin temperature can be used as an assessment tool for diagnosing and follow-up of autonomic nerve impairment. So here, I would show the mobile this infrared camera that we've used, and Android phone, which can be attached, and we can take infrared picture or a video. So our research question was autonomic nerve impairment. Can it be detected using infrared thermography in those patients in early phases of disease, which they might not show clinical signs and symptoms? Our general objective was to determine the value of IRT, the infrared thermography, in assessment of autonomic nerve function in newly diagnosed patients with leprosy. So till now, the project progress has been we've come a long way from training and setup and doing pilot patients and preliminary results, which I presented last year in spring meeting in Holland. We've done reliability tests of different cameras. We've got 3 or 4 cameras, and we did repeatability test in the same subject for number of days, and reading of those tests by different assessment by different persons. So to test the reliability of the tests, we've recruited patients, newly diagnosed patients, 2 centers of INF, one here in Green Pastures, one in the west of Nepal, a place called Nepalganj. And right now we are finishing the recruitment, and the next stage is for analysis and results and publications. So methods, our plan was to recruit 50 to 100 patients in each group. It's a cross-sectional observational study, and basically we measure the skin temperature using the IRT, the infrared camera. We get the baseline temperature of hands, palm of hands, and then the hands are dipped into cold water, the cold pressure test, and after dipping for a minute, it's again back to recording and see the temperature recovery for 15 minutes. So right now we've recruited fairly 70 patients, new diagnosed patients, and we're still recruiting some healthy controls, trying to match the age group. Just basically showing the steps what we are doing. The hands are kept still, and the camera is right on top, recording the video. For 20 seconds, we record the baseline temperature, and they're asked to take off the hands and dip into 4 degrees of cold water for a minute, which is okay for most of the patients. It's a bit of discomfort, and it's wiped, and then again it's kept back to the mold, and it's recorded for another 15 minutes. So these are a couple of significant pictures that we've seen. That's as we've seen in a leprosy diagnosed patients, which the left hand, you see the ulnar side with low temperature compared to the median and the right side. So this is the healthy subject, which seems equal temperature of both hands. After dipping in cold water, patients after cold pressure test, and the healthy, so similar results. And the recovery time for the affected side is, you can see the change. As in healthy, it recovers fairly well. So this was the response curve that we were expecting at the start of the project. The healthy would have good response, quick response. The patient with affected nerve or affected side would have this slow response. We did the infrared camera and touch temperature sensor comparison, which is fairly equal. And we did a repeatability test on subjects for 5 consecutive days, which is kind of disturbing. And some of the patients which might have, so this particular red line must be me, who was recruited as a healthy subject in this test. For a couple of days, I was exercising. I had good response, even dipping in cold water. The temperature response curve was fairly quick. So, and these preliminary results, we've not included many controls here, where we compared healthy and patients' response. The cluster of black ones are the healthy ones, and the scattered ones are the leprosy affected ones. This, for clarification, we need to have more patients who's recruited. We need to plot more subjects in this this curve. So even this is more disturbing. As you can see, it's for first time when a friend who was working on this showed me this, it felt like it's like a nerve fibers to me. So it's the black ones on top is healthy one, and the bottom one is the red ones. As you all can see, it's fairly not so much difference in these two groups. So it needs detailed analysis, particular region of interest. Are we looking at the ulnar side or median side? So this have to be analyzed again. So our learning and
We're
challenges.
almost at time, but it looks like you're wrapping up.
Yeah, I'm wrapping up.
Okay,
Yeah,
great.
be quick, very quick. So it's been a complex result. For now, it's kind of a confusion mode for me right now. It's and more data analysis and interpretation would help this. Another challenge has been this movement of hands when I try to record a video, and so we're thinking in future the AI tool for video analysis. Even there's movement of hand, we can just get the particular point with AI tool. So I've presented in a couple of local conferences, shared with the university students and visitors about this this work. There are 2 articles been submitted for publication right now. It's a technical article in a literature review. 3 abstracts been submitted to ILC 2025. So yeah, next step is research work, dissemination, publication. So I'd like to acknowledge TU Delft, the students who've helped in the studies, and team in 2 centers. Thank you.
Thank you very much.
Thank you. Very interesting results. Of course, there's immediately a question online about the results, and also for myself, it's a bit of is the person affected? Is the problem then that they don't have that particular nerve involved, or is the problem that the person not infected, so the healthy control has an issue that we don't know about? So which way does it actually go, do you think? Or is it completely random?
To just see, I've till now I've seen a couple of patients who've come with VMT, ST, or normal, with no such issues, with no such complaints of neuropathy, has shown significant results. So I would request to share one of the videos, which I'm sure you can see the difference. I quickly share this video.
Yes, go ahead.
So this is the patient who came recently diagnosed in Saudi Arabia by one of the dermatologists with a skin patch who he had for six months. So when he came here, he presented with this hand. It said, can you see this video?
Yes.
So the right hand, the ulnar side was suspicious compared to the left for me. So this is the baseline video. So if I can show the other video, which is after cold pressure test. So just a second. So this is right after, immediately after dipping cold water for a minute. So it's both hands are black, the fingers are black, it's all cold. So slowly the response you see is the palm goes first, and it starts with the tip of the fingers. And the response here, which I suspect, the median size is okay, especially the ulnar 1.5 on the right. So these are a couple of typical videos or pictures I've seen in even in early diagnosed case of leprosy. without skin smear positive.
Okay. So is this, when you do this test on people, is it accompanied by other testing of nerve function? So do you do nerve conduction testing or any other testing just to be able to compare the results or to actually identify issues with certain particular nerves in one hand or the other with other methods?
Unfortunately, we haven't included any NCV or electrophysiological tests, but we've done a complete VMT, voluntary muscle test and sensitive test for these. In further studies, it'd be really well to combine a couple of other assessment like neurophysiological studies or NCVs. To correlate what we've seen with IRT.
There's quite a lot of questions coming in, and I think they're all revolving around the same issues, of course. There's a question from Ben Naafs asking, Shetty showed that nerves from contacts can be involved. So have you been able to test not clinical leprosy patients, but contacts of leprosy patients in a similar way?
In this studies, we're basically getting leprosy diagnosed, but I have a couple of contacts I've tested, like close contacts. Like if a husband comes, then wife. So yeah. I've tried to include these couple of patients. But it's you need a large number of volume of patients for that. So which can open doors with this test. I don't know which can bring other tests, like even I was thinking about addition of nerve ultrasound to this IRT, which can be more specific if you add couple of tests to this.
Yeah. Okay, thank you. It's just generating a lot of interest, and maybe you are disappointed a bit maybe about the results, but I think it's actually helping to obtain a lot of insight on what is going on in the peripheral nerves of both affected people, but also in general. So well done.
But Professor Yan, I quickly share one of the healthy video, which might I think which is interesting, and we can see the difference. So this is one of the healthy patients, healthy controls. So this is the baseline of a healthy control, and after dipping it, it's black or cold. So response in two of the hands are quite similar. So this is so it's the recovery is exact similar in these two hands, which might differ in a lot of cases, but yeah, this is what distinct difference between these affected and the healthy controls.
Yeah. Okay. Well, thank you. I think I encourage people who still have outstanding questions to contact you directly, because
thank you.
It's generating a lot of interest. So thank you very much.
Thank you so much. Yeah.
Thank you, Dr. Maharjan. Our next speaker is Dr. Suraj Sunitha. Dr. Sunitha, if you could share your presentation. Dr. Sunitha is from the Nirikshana AIDS Care Education and Training.
Thank you, chairpersons, and good morning or good afternoon or good evening, depending on which part of the world you are. It's my pleasure to share about the use of handheld ultrasound for the early detection of leprosy and comparing it to an existing diagnostic test, which is the conventional ultrasound. So this work is mainly done in it's done in Hyderabad as well as in Lalgarh Leprosy Hospital in Nepal. And the lead applicant is Dr. Sandeep Chaitanya. We are just about in the middle of the project, 1 year into it, and this is an update of 1 year. We all know that nerve enlargement is a diagnostic criteria for leprosy, but palpation is very subjective and is a disappearing skill. Whereas high resolution ultrasound of nerves is a useful tool to objectively detect nerve involvement or nerve enlargement in leprosy. Conventionally, standalone machines are used in general practice, but the development of the handheld instrument, the Philips Lumify, has the potential of becoming a point-of-care tool to detect nerve enlargement. The study was designed to compare the USG findings in the standalone with the portable unit. India carries the highest load of leprosy in the world, and Nepal contributes significant new cases each year, and this study is set in these two contexts. Subjects, like I said, were recruited at Hyderabad and at Lalgarh in Nepal. The groups tested were newly diagnosed leprosy patients, suspects of leprosy, and normal controls. The nerves studied were 8: ulnar, median, lateral popliteal, and the posterior tibial nerves bilaterally. The parameters studied was, while doing the ultrasound, was looking at the cross-sectional area, the nerve echotexture, and blood flow on color Doppler. And the results obtained by 2 independent observers, both at Nepal and in Hyderabad. We had 2 independent observers handling the conventional machine and the handheld machine. At Hyderabad, there were 18 newly diagnosed leprosy patients enrolled, 4 leprosy suspects, and 8 normal controls. At Lalgarh, 15 newly diagnosed leprosy patients. Initial analysis indicates there is significant concurrence in the values of all 8 nerves, and in the context of both measurement, echotexture, and blood flow between the 2 machines. And so these are just some sample images using, in both these images are from Philips Lumify. On the left is left ulnar nerve, which is just 5 square millimeters. On the right would be about 26 square millimeters of another patient. Now, comparing the 2 machines here, on the left is the Philips Lumify with an area of the ulnar nerve being around 17 square millimeters, and here about 16 square millimeters on the right side. The area that is shows area there, area 1, so that is what it is. This again, if you're comparing a slightly larger nerve, when the ulnar nerve is enlarged, here we see with the Philips Lumify, it is 44, and with the standalone, it is 41 square millimeters. And blood flow, you can see here in the color Doppler on the left, there is blood flow within the nerve, increased blood flow within the nerve and also in the epineurium. And it's also demonstrated here in the conventional machine with blood flow within the nerve, in the epineurium, and also around the nerve. So these are the 18 subjects of Hyderabad. And when we studied the first one is the conventional machine, and the second column is the handheld Philips ultrasound. And you see that there is good, a paired t-test was carried out, and there was good agreement between the 2. And because the standalone was being done by one person, usually the doctor, and the technician was handling the handheld, technician trained in these techniques, there was good inter-observer reliability. This is for the leprosy suspects. And similarly, there was good agreement. And this is for the controls. Similarly, when we looked at the echo texture, it was a four-point scale of normal echo texture, mild changes, moderate changes, and severe changes in echo texture of the nerve depending on involvement. And whereas for blood flow, it was either a yes or a no. And so this is the reliability between the comparison between the two machines. These are the 15 patients tested at Lalgarh in Nepal. Again, there was good agreement between the conventional standalone machine and the Philips Lumify handheld ultrasound. These were the sample size initially planned, and out of 40 in Hyderabad, we have achieved 18 and 822 to be done. Among new patients, suspects, we have only 5 more to be done. Controls, we have 8. And Nepal, I'll come in the next slide, I'll explain the delay in Nepal. And so there are 15 new leprosy patients recruited in Nepal. So in conclusion, there were the initial analysis indicates that the handheld ultrasound is comparable to the standalone machine in clarity, resolution, in order to assess both the cross-sectional area and well visualize the echo texture and look for blood flow using the color Doppler. And with ease of operation, it's nice to see the technicians trained both at Nepal and also in Hyderabad, handling the handheld ultrasound, the Philips Lumify, which so well. And we want to thank Dr. Suman Jain and Dr. Leo Visser, who trained both at Hyderabad, and Dr. Suman Jain trained also at Nepal, the doctors and the technicians. So the machine reliability and inter-observer reliability has been good. We will continue to look at intra-observer reliability, which will be carried out soon. So the handheld ultrasound is a useful, reliable, and versatile tool for the diagnosis of leprosy at point of care. A few limitations: there was a delay in ethical approval, and Dr. Suman had a lot of international travel, and so the training was delayed, but we are confident, considering the number of patients seen both at Nepal and also in Hyderabad, that we'll be able to achieve the numbers within the time frame of the study. A number of normal and leprosy patients continue to be recruited, but a lot of the suspects that are referred with sensory motor changes turn out to be pure neural leprosy or determined forms of leprosy, and so the suspects is lower. The future plan is to complete the sample numbers and correlate the findings with other clinical and diagnostic parameters. This was mainly a study that compared the two machines to see whether they were both agreeing with each other. We want to thank the Turing Foundation, who co-funded the research project through LRI, and to the patients, suspects, and controls who willingly participated in the study, and to all those who helped in the study, both at Nepal, a very committed team at Nepal and a very committed team in Hyderabad. A big thanks to them. And to Hope Rises, formerly ALM, for support at every stage. Thank you.
Thank you.
So we don't have any questions as yet, so I encourage participants to submit questions. I have one question for you, quite general. If you have completed the study with more participants or more controls, and it comes out more or less as you have shown in this presentation. What would be your recommendation? What kind of recommendation would come out of your study?
Thanks for the question, Richard. I think more and more people are learning to use the ultrasound, and it is a very good technique to apply, very much so in pure neural leprosy, where people come with peripheral neuropathy and no skin lesions, but also in other forms of leprosy. It is very useful to establish without doubt the enlargement of nerves, and so I think it is something that should be used as an ancillary auxiliary tool in the diagnosis of leprosy. Yes, nerve palpation, sensory testing, voluntary muscle testing, but also the use of an ultrasound is very, very valuable. And we have used it for many years in Hyderabad, and we find it very useful. I hope I answered your question, Richard.
I think so. Basically, you're recommending the application of the new apparatus. I think that's what you're saying. Yeah.
Yes, and that this particular handheld tool attached to even a mobile phone or even an iPad is, I mean, it is a point of care value.
Yeah, absolutely. We have some questions coming in now. One is, will you later try to correlate palpation results with the HUS results?
Say that again. What results with?
Are you planning to correlate afterwards results of palpation with your handheld ultrasound?
Yes, of course. So we have the details for all these patients. The data collection tool includes nerve palpation, sensory testing, and voluntary muscle testing for each of these patients, and also number of lesions. So everything will be correlated at the end of the study.
Yeah, so in the end, you will bring all this data together for correlation testing. Thank you. Then the next question is about the fact that nerve sizes can change with time of day, with individual activity, or even hydration. So if this is so, how do you take that into account? How would this impact your study results?
Yeah, definitely. At diagnosis, I mean, it would be used, ultrasound would be used to detect nerve enlargement at diagnosis. But if there is a type 1 reaction or there is clinical neuritis, pain, or tenderness, we would also look at the nerve. At that time, there is definitely a chance that the enlargement of the nerve could be even more because of the neural edema. And therefore, we can look at that. And then during follow-up, you can, with treatment with steroids, you can also see a slight reduction in the size of the nerve and a reduction in blood flow as an indication that the neuritis is reducing. So you can study the nerve over time and look at changes in the cross-sectional area as well as the internal structure of a nerve.
Yeah, and from what I remember from my own experience long ago, the nerve size can also be dependent on if a person is involved in manual labor and such kind of effects. So I don't know if this is something that you take into account as well, that there can be individual differences in the size of certain nerves.
Yes, definitely. There would be a slightly larger right ulnar nerve compared to the left in manual labor and people who use it. And we are also going to study differences between ethnic groups like Nepal versus India. And we might see some differences between the cohort at Nepal and at Hyderabad. So yes, we will be looking at those.
We have one final question, very down to earth. What is the cost of the machine?
It's very reasonable, Richard. I mean, it costs about 5,000 USD, the Philips Lumify, and maybe with an iPad and maybe 6,000 USD. So I think it's much less than what you would pay for a standalone machine. In India, a standalone machine will be about 20,000 dollars. So
Right.
$5,000 to $6,000 is definitely much better.
Okay, thank you very much for your presentation. I'll give the chair back to Kate.
Thank
Thank
you.
you.
Thank you, Dr. Sunitha, for your presentation, and thank you to all of our presenters. And I'm so happy that we had such engagement, even for a Zoom presentation. I see 28 answered questions in the Q&A, which is really exciting. We are technically out of time. So I will close the session and say thank you to everyone. Exciting results. Looking forward to seeing what happens in the next year, and seeing you in the sessions tomorrow. Dr. Richardis, did you want to add anything?
Thank you all for joining. We had lots of participants. I think there were 75 at the height of the meeting. So very good.
Yeah, very exciting. Thank you, everyone, and have a good morning, afternoon, evening, all across the world. And my best to you all. Thank you.